Hello, am I audible? People? Mariam?
Yes, sir.
Yes. Okay. Can you see the notebook that I'm sharing?
Yes, sir.
Aisha, what were you saying? Aisha, are you there? Let's start a new page, shall we? Aisha, are you there? We're still waiting for you. Okay, she is reconnecting probably. Mariam, you have any doubts?
No, sir.
Oh, everything is clear? Just give me one moment. Don't know. Okay, let us start a new page. Okay, so it's visible to all of you, to both of you. I think Aisha is rejoining. Mariam and Riva.
Yes, sir.
Okay, so in today's class, so in the last weekend we finished aerobic respiration, and you all are fine with it. Everything is clear with aerobic respiration. How many total number of ATPs did we manage to synthesize out of one glucose molecule in one complete? No, in one complete cycle of aerobic respiration from glucose till CO2, how many ATPs did we generate total in one aerobic respiratory cycle? Mariam, shall I go to the... Okay, can you see this?
Yes, sir.
So was the final from glucose till we get CO2 in the end. See here, the only thing which is not energy intermediate or an energy molecule is CO2, correct?
Yes, sir.
So basically we started with glucose and we ended at, oh, what's happening? Just 1 minute. Yes. We started with glucose and we ended at CO2. Okay. Is my voice clear to all of you?
Yes, sir.
Okay, the pen was not working. Okay. So glucose to CO2, everything else, ATP, NADH, and FADH2 are energy intermediate. So this is the energy currency. So this is one full aerobic respiratory pathway. Okay. Now, how many ATPs did we manage to make finally? 38. Is it clear, Mariam? Not 2.
Okay, sir.
So the question was one full aerobic. In one aerobic cycle, from one glucose, we managed to make 38 ATPs. Okay. And this was the electron transport system. And I told you about the complexes. Everything is clear, right? Any doubt in this complexes? And I also asked you to go and look at ATP synthase. Did you do that? This big motor-like molecule, ATP synthase. It's very beautiful molecule. Now let's come to today's topic. So today I'll be completing what's left in the respiratory pathway and the respiratory chapter. And most of it is actually your respiratory quotient. And one important topic is fermentation. So what is fermentation? Let's begin with fermentation. Mariam, any idea what is fermentation?
Where yeast forms ethanol
Just ethanol?
and energy.
But what is fermentation? We can form ethanol in beakers also in a chemical reaction. What is fermentation? Is it something, has it something to do with the respiration? Why are we studying it here? So what was happening in the aerobic respiration that we studied? A complete oxidation of glucose was happening, correct?
Yes, sir.
In fermentation, it's again a pathway by which energy is produced in cells, but under oxygen limited environment. Okay. Is it clear?
Yes, sir.
Fermentation. Is fermentation an anaerobic mode?
Yes.
No, it's not exactly anaerobic. So what is anaerobic? Before we start understanding fermentation, let's understand what do we mean by anaerobic. I told you. What is anaerobic? What is anaerobic respiration, to be precise?
Respiration in the absence of oxygen.
Yes, where oxygen is not at all utilized or it is not at all present. Is it clear? But fermentation is called incomplete oxidation. What it is called? Incomplete oxidation or partial oxidation. Of glucose. Is it clear? If
Yes,
it's
sir.
incomplete, but there is oxidation, correct? So oxygen is there, but it's limited in supply or it's very, very less, correct? It's not complete absence of oxygen. Now, how do we differentiate between aerobic and anaerobic respiration and fermentation? Write down certain points that are important for your entrances, because sometimes they confuse in entrances with anaerobic and fermentation. So write down anaerobic respiration is different from fermentation. Anaerobic respiration is different from fermentation. In anaerobic respiration, the final electron acceptor, remember there was an electron acceptor in aerobic also. What was electron acceptor in aerobic? The final electron acceptor in aerobic respiration. What was it? Which molecule was the final electron acceptor in an aerobic pathway? Mariam, Riba.
Oxygen.
Exactly, oxygen, O2. We gave all the electrons to O2 and make water, right? And while we were doing that, the complex 1 was pumping out protons, right? In exchange. And that proton was again finally coming back through ATP synthase, making ATPs. Remember?
Yes, sir.
Yes. So the final electron acceptor there was oxygen. In anaerobic respiration, the final electron acceptor is either nitrate, just write down these things. Either it is nitrate, it can be sulfate or fumarate. Remember fumarate, we made fumarate in Krebs cycle.
Yes, sir.
Yes. These three molecules or something which is not oxygens, something which is other than oxygen. So you can also write down a compound other than oxygen. And it's clear now, if it is oxygen, then it is aerobic respiration. If oxygen is not present, but still you have to make energy, of course you will make less energy, but you have to make energy. So you have to use another electron acceptor, which is not oxygen. So which is nitrate, sulfate, or fumarate. So that is anaerobic respiration. Whereas in fermentation, write down, in fermentation, the electron acceptor is the final electron acceptor. Always write final, because remember in that electron transport chain, there were many molecules which were taking electron and passing it, right?
Yes, sir.
They were all intermediate electron acceptors. One was the Q molecule, remember? The UBQ-none. And then it passes to complex 3. In the photosystem, in photosynthetic plants, in the photosystem, there were other electron acceptors. Is it clear? Final
Yes, sir.
acceptor there was NAD+, which was taking that electron and becoming NADH. Remember the photosystems, the Z scheme? I hope you are all revising those things. Okay. In fermentation, the final electron acceptor is, write down, an endogenous organic molecule. Final electron acceptor is, it's not nitrate, it's not a charged, it's not sulfate, fumarate, it's an endogenous. Endogenous means which is present already inside the cell in the cytoplasm. Endogenous, and it is an organic molecule. It's not inorganic. Like nitrate is inorganic, correct? NO3. Oxygen is also inorganic. We take it from atmosphere in elemental form, right? But organic molecule is which is something which is inside already. For example, like you gave an example, remember Mariam? Lactic acid or ethanol, correct?
Yes, sir.
So in fermentation, the final electron acceptor is a endogenous organic molecule and not this. That's why it's different from anaerobic respiration. Now write down another thing. Fermentation is a self-contained process. Write down this point. Fermentation is a self-contained process. This simply means that there is no electron acceptor which comes from outside, which the cell needs from outside. So it's like self-sufficient. A cell is self-sufficient to do fermentation under low availability of oxygen. Okay. What else? It does not require an electron transport system. Write down. Keep writing these points in fermentation. It does not require or involves. It does not involves an electron transport system. Okay. And no ATP is synthesized. Next point. No ATP is synthesized by oxidative phosphorylation. Make sense? Everything makes sense?
Yes, sir.
Yes. Now, when does fermentation takes place? So in any cell, remember the universal process with which energy formation starts is glycolysis. That is true for all living cells on the planet. Is it clear? So it starts with glycolysis. And how many ATPs do we produce in glycolysis?
2.
2 net. Okay, 2 molecules. And everything starts after that. So fermentation also starts after glycolysis, which means what will be the starting product for fermentation?
2 ATP.
No. The starting initial reactant, not product, the initial substance with which fermentation will begin. What was the end product of glycolysis?
Pyruvic acid.
Perfect. So you will call it pyruvate from now. Remember why? Because I will tell you why it's important. Because when we call something acid, we are telling that it is in the acidic form in the cell, but it's not. Pyruvic acid stays as pyruvate in the cell. Remember?
Yes, sir.
Carbonic acid was carbonate. in the cell. Similarly, so pyruvate is the end product. You are correct. Now, so what should be the first product, the first compound with which fermentation should begin? Pyruvate. Okay. Now, why does fermentation happen? So basically, what I have already told you is, if pyruvate cannot go into mitochondria, if pyruvate goes into mitochondria, what will happen? Pyruvate will become, come on, I'm just repeating all this. I'll keep repeating till it's set in your head in stone. Okay, let me use blue to say where this thing is happening. So in cytosol, we have a molecule called pyruvate, which came from glucose. Okay, so this pyruvate, if it goes in mitochondrial matrix, then what will happen? Inside mitochondrial matrix, pyruvate will become acetyl-CoA. Remember?
Yes, sir.
And where will this acetyl-CoA go? In the Krebs cycle. Remember?
Yes, sir.
And we will get NADH and FADH2 from the Krebs cycle, which will go into the electron transport chain and will finally make water out of O2 and lots of ATP, exactly 38 in total. Is it clear?
Yes, sir.
Now, if this thing is not possible, and suppose it has less oxygen, because whenever it was going this way, in the Krebs cycle, what we are generating lots and lots of NADH and FADH2, which requires oxidative phosphorylation to produce energy. Is it clear? So if we are committing to Krebs cycle, if a cell is committing to Krebs cycle, it has to ensure the supply of oxygen, correct or not?
Yes, sir.
Otherwise it's waste, because all you will get from Krebs cycle is lots of NADH and lots of FADH2. But if you cannot oxidatively if you cannot do phosphorylation, oxidative phosphorylation of them, you will not get ATP. Do you understand? So if whenever there is very less oxygen available, pyruvate, instead of going into mitochondrial matrix and going through this pathway to make lots of ATPs, it undergoes fermentation. Is it clear?
Yes, sir.
Yes. And this fermentation can be of two types. One is called lactic acid fermentation, and the other is called ethanol fermentation, or in other words called alcoholic fermentation. Okay, clear? Now let me ask you a question. Where have you heard this word before? Lactic acid.
Muscle cells.
Yes. When Mariam?
When we do vigorous exercise.
Exactly. So what happens when you do vigorous exercise? What do you think?
We need lots of energy.
What?
We need
You need, yes, you need lots of energy. So so does fermentation happens in your cells, in your muscle cells? Do you think to make lots of energy your muscle cells are doing fermentation? They can simply go through this pathway in Krebs cycle, right? What do you think is happening? Another factor that you just missed, when you are vigorously exercising, what's happening to your physiology? Think, it's all biology, it makes sense. Suppose you are playing football. What sport do you play, Mariam? What's your favorite sport?
throw ball.
Throw ball.
Yes, sir.
Snowball. Throw ball.
Throw ball.
Yeah, nice. So when you're playing throw ball, let's say you are playing throw ball vigorously, it's a very competitive tight match and you're playing throw ball. So what's happening to your physiology? When we are doing anything vigorous, when we are running, jumping, playing throw ball, football, can we breathe properly? Are we breathing?
No.
No. How are we breathing then?
We are breathing, but not sufficiently,
Sufficient in the sense of what?
not calmly.
Calmly is like, because you're sitting, you're calm. So when we run, when we start running, our breathing rate increases. We take deep breaths like this, right?
Yes, sir.
And why does our body do that? It's a very simple logic, because we, when we were sitting calmly, what you were saying, breathing calmly, you are not utilizing lots of energy, right? All of a sudden you started playing throw ball, and your muscles, all your legs, all the part of your body, your heart is just expending energy like mad. And your brain tells your lungs that this person has just gone mad. She has started playing throw ball. So let us work now. Let us increase the rate of energy formation. But in order to do that, when you are not able to bring in sufficient oxygen required to burn all that glucose which is there, you understand? So what happens is, whenever there is vigorous activity, there is less oxygen which reaches our muscles. Why less oxygen reaches our muscles? How do oxygen reaches our muscles? Through
Diffusion.
blood.
Yeah, blood.
It reaches through blood. And muscles, before reaching the muscles, all our brain, heart, and every organ is also using oxygen a lot. So a lot of oxygen gets used up in the journey, and very less oxygen reaches the muscle for that to sustain that vigorous activities like gymming, playing vigorously throw ball or something like that. That's why after, if someone is not in a habit of playing regularly, and someone goes and starts playing throw ball or football and keeps playing for 1 and 2 hours, so the body starts to ache, right? Same with the case of gymming, right? It starts to ache. Muscles start to ache. Basically there is a pain in muscles, correct? And that
Yes,
pain
sir.
is because of formation of lactic acid in the muscle cells. And that happens because lots of oxygen is not available. So whatever oxygen is available in to use that oxygen, and at least partially like partially oxidized glucose, cells, instead of going through the aerobic pathway, they starts going into fermentation. Is it clear?
Yes, sir.
And lactic acid fermentation happens in our muscles, and that's why our muscles hurt after a vigorous activity. The second is ethanol or alcoholic fermentation. Where have you seen this happening? So the example of this is your muscles after a muscles, not after, during a vigorous activity. Okay, where does this happen?
Yeast.
Yes, yeasts. Alcohol formation, right? Remember, alcohol is produced through fermentation. Fermentation of rice, grapes, sugarcane leads to alcohol formation. So basically yeast makes this ethanol which is there in alcohol, okay, as a beverage. Now yeast, under oxidative stress and under less oxygen, will keep on making ethanol. And both of these products, are they good for the cell, lactic acid and ethanol?
No.
They both are harmful for the cell in excess. Okay, ethanol, yeasts cannot tolerate more than 13% ethanol concentration. So at 13% ethanol concentration, they die. Okay, so yeasts die at 13%. So whenever in yeast, suppose they are continuously doing alcoholic fermentation, when the level will reach 13%, they will die. So if I tell you, using yeasts, what is the maximum concentration of ethanol you can reach in any beverage or food?
16.
13. 13 to, where did you read 16?
Sir, I remember one of my friends saying that like 15 to 16.
It's actually 13. Okay. So at 13, if there is a range given, then it should be 13 to 16 in some books. But remember that at 13%, they starts to die. At 16, all of the yeasts will die for sure. So even if you're taking a range, which is 13 to 16, let's say, okay. What is the maximum amount of alcohol that can be present in any naturally fermenting beverage?
13.
13, right? But we do get alcohols which are more concentrated than 13%. Do you know that?
Yes, sir.
You must have seen your hand sanitizers. What is the alcohol concentration in that? 70% ethanol. How can we ever make 70% ethanol or 99% ethanol? Or in a alcoholic beverage, still we can go beyond 13% in some like vodka or absolute vodkas. We get more than 25-30% of ethanol concentration. So how do you think we get that? Any idea? Biologically and scientifically, how is it possible?
Sir, can we mix only 13%?
13 plus 13 becomes 26, right? Okay. So I leave that question to you. I'll answer it, but not today. Okay. Let me first go a little bit into the chapter. But if I could not answer it today, then it's your assignment for tomorrow. You have to answer it before I start the class tomorrow. So write down this. How can we make concentrated alcoholic beverages beyond the threshold of yeasts, which is like 13 to 16%? Written?
Yes, sir.
Yes. Okay, so let's start with the first type of fermentation. Let's start with lactic acid fermentation first. So even if these things are not given in detail in the NCERT, but they are important for entrances. So there is a I think there is a flow chart given in NCERT for fermentation, and that flow chart summarizes the fermentation totally. We'll come to that after going into the detail. Okay. So let's start with lactic acid fermentation, which happens in our muscles. Okay, now write down. Apart from muscles, can you tell me any other place or any other substance where you have heard this word lactic acid? How many of you like curd?
Yes, sir. Lactose.
Yes. So lactose is a sugar which gets converted into lactic acid by lactobacillus bacteria, right? That's called curdling of milk. So curdling of milk, in this process, we actually are doing lactic acid fermentation. Just like glucose is a sugar, correct? Fructose is also a sugar. Similarly, lactose is there in your milk. So, and what is lactose? Lactose. Find out the monomers of lactose, okay? So lactose gets converted into lactic acid. And what happens? Why do you think liquid milk becomes solid curd? Any idea? Any logic? This is a question, by the way. It's a board question. What's happening to the milk when it's becoming curd? Explain the process in detail. 2 marker, 3 marker, 5 marker. So we know that lactose is being converted to lactic acid by a bacteria called lactobacillus, right? The word bacillus gives the shape of the bacteria. Bacillus means they are rod shaped. They are like this, bacilli. The bacteria looks like this. So this means bacillus. And because they convert lactose to lactic acid, they're called lactobacillus. They feed on lactose. So this we know is happening. Now this is an acid, right? So what's happening to the milk? Before there was no acid, there was a sugar. So this is a sugar in the milk. Now there is an acid in the milk. So what happens in the presence of acid? Milk has lots of proteins, correct? Any protein in the milk that you can name, Mariam? Any milk protein? Any example of milk protein, please? Milk has lots of protein. One is casein. Okay. Remember this name, casein. Okay.
Yes, sir.
Now proteins, whenever proteins interact with acid, they solidify. Plus acid plus protein, it becomes solid. Proteins become solid. And this is exactly what is happening in the milk. So when you introduce, and you must be knowing that to make a milk, convert milk into curd, we have to put an inoculum into the milk, right?
Yes.
At a right temperature. How many of you have seen your mother's curdling milk in your house? Are you familiar with it, Mariam?
Yes, sir.
So what does your mother do? Can you take me through that process? What does she do? Let's figure out what science she's doing, even if she's unaware of it. So you can go back and tell her that, see, this is the science you are doing. Basically, kitchen is just a laboratory. Lot of science is done in kitchen. I always tell my mother that, there are two scientists in the house. She is the practical one, I'm the theoretical one. So she does all that thing which I study. So what does she do? What does your mother do?
Sir, I saw it on movies. In movies.
Okay. What have you seen?
A pot and then milk inside it, and then they just churn or something.
No, that is to take out that what do you call makhan or ghee. Churning is churning of milk to take out makhan or ghee. Curdling of milk, it's simple. You take milk, you boil it, then you let it cool. Okay. So you're boiling milk, and you can, because milk which comes in the packets are really cool, like they are cold stored. So you boil it at home, and then you bring it to a normal temperature, and before it becomes like completely cool, you will see that your mother adding one small spoon of curd in it, and that leaving it at the room temperature. If you keep that pot with inoculum in refrigerator, the curdling process will get delayed. And you can very well understand by now why. Because whenever there is an organism involved, lactobacillus is a bacteria, it's an organism. And to do anything metabolically, it requires enzymes, correct? And enzymes work best at a particular range of temperature. If it's below that, it will not work. If it's above that, enzymes get denatured. Is it clear?
Yes, sir.
So for converting milk into curd, a temperature is required, plus other factors like humidity, other things are also there. But you will see that they introduce inoculum, we call that small part of curd inoculum, into the milk, and then they keep it untouched. So it should not be touched, because what's happening is lactobacillus first is feeding on this lactose and multiplying in number, and simultaneously converting this sugar into lactic acid through lactic acid fermentation, correct? So over time, after 6, 7, 8 hours, lots and lots of bacteria will be there, and lots of lactic acid will be there. So what this lactic acid will do, it will start reacting with all the protein which is present in the milk and start solidifying. Okay. So all this protein together clumps together, clumping everything which is there in the milk, and the white liquid becomes white solid, like a gel-like substance, which is called curd. Is it clear, everyone?
Yes,
so that's
sir.
what's happening. That is what's scientifically happening. Now let's write down the same thing, what our mothers do in kitchen. Let's just write that reaction, okay, for lactic acid fermentation. Write down first few things first. Muscle cells and certain bacteria like lactobacillus, muscle cells, and certain bacteria like lactobacillus oxidize NADH by transforming pyruvate into lactate and this is called fermentation. Okay. So what's happening basically is, so should I always start from sugar or can I start from pyruvate? If you are all okay with understanding that glucose through glycolysis, okay, let's start from the beginning only. It's good to. So glucose becomes pyruvate through which process? Just keep repeating.
Glycolysis.
And what was the energy output?
2 ATP.
2 ATP. And in terms of how many NADH we get? ATPs, let's not count ATPs because they are used. Okay, finally. So we'll get confused otherwise. So ATP is used. It's a finished currency. We can just use it. We also got some NADH in the process of glycolysis. Can you tell me how many NADH we get? NAD plus was converted to NADH. Remember in glycolysis?
1, 2.
2, right. So 2 NADH becomes 2 NAD plus becomes 2 NADH. So this is what we got. And as a byproduct, we also got 2 pyruvate, right? Now this 2 pyruvate will enter lactic acid fermentation. In our muscle cells and in lactobacillus. And what will be formed? Two lactic acid molecules. Okay. And in this process, you remember these 2 NADH? These 2 NADH are now in this process. 2 NADH goes in, and what happens here is oxidative phosphorylation because there is less oxygen, so only these 2 can get oxidatively phosphorylized and 2 NAD plus comes out. Okay. And this process is happening so whenever NADH is involved, I have told you, and we are basically removing hydrogen. So can you guess the name of the enzyme which is playing part here in lactic acid fermentation? What is the other name of lactic acid? What I can call lactic acid as? Can I call it lactate? Because to stay in the cell, it has to break, like it should be in the form of lactate, otherwise it will hurt, right?
Yes, sir.
So we can call it lactate, and the enzyme is called lactate dehydrogenase. Do you understand why it's called dehydrogenase?
Yes, sir.
Yes, because this hydrogen is getting removed. So write down, lactic acid fermentation is catalyzed by the enzyme lactate dehydrogenase. In short, it's also called LDH. Commonly, LDH, lactate dehydrogenase. Okay. Now, when this is formed in our muscle cells, so everything is clear till here?
Yes.
So when we do vigorous exercise, like Mariam played vigorously throw ball and now her muscles are aching, they're hurting, they're paining. So what happens after a few days or after some time? What happens basically? Does it keep paining forever?
It get better.
It gets better, right? Why does it get better? What do you think? Because there was lactate or lactic acid present in the muscle, so it was hurting. So what happened to that lactate? What can you think has happened?
Then oxygen was available, so
So,
muscle took the oxygen.
but even if it takes oxygen, do you think lactic acid can anywhere fit in the Krebs cycle? Have I go through the Krebs cycle? Have you heard? Did you read the name lactic acid or lactate there? There was citric acid, but not lactic acid, right?
Yes, sir.
Can we put this into the Krebs cycle now?
No.
So we have to clear it from the muscle. Remember, after, so suppose your body is aching from exercise, and then suppose you go to a doctor and say that my body is aching. Doctor says, okay, drink lots of fluid, lots of water, right? They don't give you painkillers for this because
Yes,
it will
sir.
not work. It's not a neural pain, right? It's not coming from receptors. It's your muscles have lactic acid deposited in it. That's why they are hurting, correct? Once this lactic acid is cleaned, flushed from there, it will stop hurting. So the more water you will drink, the more fluid you will take, your blood will have more and more fluid. And remember, more and more lymph will be formed. Lymph is basically the plasma serum leaking out from the capillaries, correct? And they keep cleaning the muscles, right?
Yes, sir.
Yes. So basically what happens is this lactate, which is formed in the muscles, is transported back to liver, where it is converted back into either glucose or pyruvate, both of which are useful to us, right?
Yes, sir.
Yes. So write down this, write down few points. In animals, the lactate formed in the muscles is transported back to liver to produce glucose. Okay. Now the point is, if you are producing glucose back, what can you do? You can do glycolysis. Now to produce it back, you might use some ATPs, but again you can do glycolysis and come to pyruvate. So pyruvate again is useful because you can now, under normal oxygen concentrations in your body and in your muscle, when you are calm and sitting and composed, where can you send that pyruvate? In the mitochondria to make what?
ATP.
Acetyl coenzyme A, which will go into Krebs cycle to make lots and lots of NADH and FADH2. And since, because now there is lots of oxygen available, you can oxidatively phosphorylize them and make ATPs, correct?
Yes, sir.
Perfect. So write down. In the liver, the lactate is reoxidized. The lactate is reoxidized by the liver LDH, because this process is, it goes both ways. Okay. Liver LDH means liver lactose dehydrogenase enzyme. So with the help of liver LDH, it can reoxidize it to pyruvate. The first step is from lactic acid to pyruvate. See, the path you follow to reach lactic acid, you will follow the same path back to glucose. You understand? So you cannot directly jump from lactate to glucose. From lactic acid, you have to go back to pyruvate, right? See here, this way. Is it clear? And then from
Yes, sir.
pyruvate, you have to make glucose back. So first it will reoxidize lactic acid through liver LDH into pyruvate, and then write down. So what we are doing finally here, you must have realized in the liver, when we are making glucose from pyruvate, we are synthesizing new glucose, correct? So the process is called gluconeogenesis. Is it clear, Mariam?
Yes, sir.
What does the word say? Gluco means glucose. Neo means
New
new, and genesis means synthesis.
making synthesis.
Yes, making or synthesis. So we are making new glucose, which was not there in the cell from anything. In this case, it is lactic acid. Anything means something which can be, of course, converted under the laws of chemistry. So from lactic acid, which was formed in our muscles, slowly on day 2, day 3, all that lactic acid goes back to your liver, where your liver, with the help of lactate dehydrogenase enzyme, converts it back to pyruvate, and then converts it back to glucose, and then stores it. Okay, that's called
Yes, sir.
gluconeogenesis. Write down. By the process of gluconeogenesis, pyruvate is finally converted back to glucose in the liver. And gluconeogenesis only happens in the liver, okay? So glycolysis, gluconeogenesis, two things are clear to you?
Yes, sir.
Now the third thing, there is a third similar term. So you are clear with glycolysis, this term. So see, all these terms are here. So let us discuss the third one also. This you are clear with, this you are clear with. There is another term which is called glycogenolysis. Glycogenolysis. Now let's again, what the word itself tells what it is doing. Here the word is glycogen. Glycogenolysis, two words. So basically what it tells, that you are, glycogen means you are breaking glycogen, correct?
Yes.
What is glycogen? Anyone remembers glycogen? What is glycogen?
The animal.
The stored.
Yeah, the stored form of
It's a long
glucose in
chain.
animal.
No, it's a long chain of glucose. When I was telling you about digestive system, I told you that all that
Yes, sir.
glucose, all the glucose that we get from the food, which we absorb from our intestines, basically they all go and get stored in the liver in the form of glycogen. And glycogen is nothing but a very long chain of glucose attached to each other, is called glycogen. So glycogenolysis happens when we are breaking down glycogen in the liver. Gluconeogenesis happens when we are making new glucose from lactic acid in the liver using LDH. And glycolysis happens when we are breaking glucose into pyruvate in the cell. Is it clear?
Yes, sir.
Everything is clear? No doubt.
Yes, sir.
Okay, now write down. Now what happens? Liver has made this glucose. Now it has two things it can do. If there is lots and lots of glucose, then it will convert it into glycogen, which it has already done. But this glucose, it's newly synthesized, right? So what happens? It sends this glucose back to muscles. Most of the time, sends back to muscles. Why is it sending it back to muscles? For glycolysis. Make sense or not?
Yes, sir.
It tells the muscle, see, you could not oxidize it completely because of less oxygen. Now for you, I have converted it back to glucose. Now do what you were doing. Okay. So now there is no oxygen stress or less oxygen, or you are not doing vigorous activity. So this new glucose, which is synthesized, has two options: either to get stored as glycogen, which the liver is already doing when we are eating things. When we are eating lots, every human eats more than what is required, because hunger is psychological. You know that? More than physiological. Hunger, the feeling of hunger or the feeling of eating, is psychological. So we all have a habit, a circadian rhythm of doing breakfast, lunch, and dinner, correct? And we keep doing it. So even if, let's say, whatever amount of energy a human needs throughout the day, if I just can purify glucose and can fit all that glucose required, pure glucose, in a capsule, and I ask you just take 3 capsules in a day, and you have nothing to eat, will you be okay with that? Will a human be okay and not feel hungry?
No.
No, because the system is built like our stomach. When it will not get something at 2 o'clock in the afternoon, it will start producing more acids, right? And you will feel hungry. You want
Yes.
to eat. So we eat to fill our stomach. We do not eat just to suffice our need of energy. Is it clear? And that is how we are shaped. So we always have reserves. That's why a human without food can sustain for around a week without eating anything. It will not die, because in that case, what will happen? Glycogenolysis will happen. Glycolysis will also happen. Gluconeogenesis may not happen, but glycogenolysis is happening, and we get back all that reserves from our liver. So why I started this is, yes. So basically, we have glycogen stored. So this new glucose that we synthesize through gluconeogenesis, we send it back to muscles for glycolysis. B means liver sends it back to muscles. Everything is clear till now?
Yes, sir.
Very good. Now write down this whole pathway. This whole pathway. Let's make this pathway. This is a very small. It's not actually a like it can be called a pathway because it's in a cycle. So how should I make this? Let's say on one side you have your muscles. So this area is your muscles. Okay. The skeletal muscles mostly. See, we have two types of muscles, the skeletal one and the smooth muscles, which are present in each and every organ of ours, right? When we are exercising, we are not using the muscles of our stomach. We are using the muscles of our body, the skeletal, which we use to move, correct? Do you understand the difference between the two? Both of you?
Yes, sir.
Yes. So skeletal muscles, suppose this is skeletal muscles, and then we have this area connecting. And finally, this is your liver. What is connecting muscles with liver? Of course, one thing connects everything in the body. And what is that?
Blood.
Yes. So in the skeletal muscles, what happened? We got glucose, and through glycolysis, this is the pathway, this is the cycle. Okay, it has a name. It is never asked in boards, but has been asked in entrances, so you should know it. Glucose converts into pyruvate, and if under some oxygen stress, pyruvate gets converted into lactate or lactic acid, then it has to be removed, correct? Now 2 pyruvate and 2 lactic acid, just to keep it. So this lactate is taken into the blood, and from blood it goes into the liver. Is it clear? Where this lactate is again converted into pyruvate. And which enzyme is used here? Liver LDH. So LDH of liver, lactate dehydrogenase. And this pyruvate is again converted back to glucose. Okay, 1 glucose. And what is this process called? Gluconeogenesis. What I'm writing is clear? Is my handwriting okay?
Yes, sir.
And then this glucose is taken back by the blood and sent back to muscles. So do you see there is a cycle happening? If you are in a regular habit of doing exercises, working out, doing playing something, then this cycle is happening regularly in your body, correct?
Yes, sir.
So this cycle is called the Cori cycle. It was discovered by Cori. It's called Cori cycle. It has another name. It's also called lactic acid cycle. Remember, citric acid cycle is different and lactic acid cycle is different. Okay, citric acid cycle is Krebs cycle. Lactic acid cycle is Cori cycle. Okay, just make this
Yes, sir.
and remember this. It is important for entrance only. I don't think it's for your boards. So I think that's all about and one thing about this cycle is this person got Nobel for figuring out this cycle. Nobel Prize in Medicine and Physiology. You know, there is no Nobel for biology. You know that? Because what we study in biology, till it has any practical implication, we cannot give a Nobel for it. So Nobel Prize in Medicine and Physiology is there. People call it the Nobel Prize for biology, but it's not actually for biology. Similarly, there is no Nobel for theoretical physics. Okay. And because mathematics is all theoretical and practical implications of it comes later, there is no Nobel for mathematics. So there is Nobel for chemistry, medicine and physiology, physics, economics, and peace. Okay, just something you should know. And this cycle was the Nobel they got was in the year India got independence. So that's how I remember it, 1947. They got Nobel. Okay. Just write it if clear, everyone.
Yes, sir.
Okay, cool. So I think that's all you need to know about the lactic acid fermentation. Let's start with the second one, which is your alcoholic fermentation. Alcoholic fermentation. Now, write down few things about alcoholic fermentation first. Just like lactic acid fermentation was happening in our muscle cells and certain species of bacteria, write down alcoholic fermentation occurs. Alcoholic fermentation occurs in yeast, and some bacterial species. Alcoholic fermentation occurs in yeast and some bacterial species. Now, as we know what's happening here, again, we are starting from pyruvate, but that pyruvate, instead of getting converted into lactic acid, is now being converted into ethanol. Okay. Okay.
Yes, sir.
Now that is a two-step process. Like here, it was a one-step process. From pyruvate to lactic acid is a one-step process, right? You have this enzyme called LDH, and this enzyme just can convert pyruvate into lactic acid. Simple, clear, one-step process. Write down alcoholic fermentation is a two-step pathway. So I always start the pathway with glucose, but remember that glucose is not a part of this pathway, right? because from glucose to pyruvate, it's a separate pathway called glycolysis. I hope you all are understanding, and it's obvious, right? So I will not use glucose now. Or should I use it? Because I have to show NADH now. Let's use. So since I'm again using glucose, but remember, this is not a part of alcoholic fermentation. So from glucose to pyruvate, which is 2 in number, happens through same thing, glycolysis. And what do we get? 2 NAD plus enters, and we get 2 NADH from glycolysis, correct? For this, I have to show this. Now, pathway alcoholic fermentation starts from here. Now, this pyruvate, which is 2 pyruvates, first, write down, converts into acetaldehyde. Acetaldehyde. Now, what is acetaldehyde? It's aldehyde. It's clearly like the functional group is aldehyde, right? So we are converting an acid. First, we converted a sugar to acid, a pyruvate, and then an acid to aldehyde. So can anyone guess the end? When we are doing this, we are losing 1 carbon from each pyruvate in the form of CO2. So which means there are 2 pyruvates, so there will be 2 CO2s which we are losing. So basically we are removing 1 carbon. So if pyruvate is a 3 carbon molecule, acetaldehyde will be a
2 carbon.
Yes, it's a 2 carbon molecule. So I'm writing it 2C molecule. Remember this. Because 1 carbon is getting lost in CO2. Now, if we are losing carbon, losing CO2, so when we are remove carbon, we call it decarboxylation. We are doing decarboxylation. We are removing carbon, just like dehydrogenation. So for dehydrogenation, we use the enzyme dehydrogenase. For decarboxylation, what will be the name of the enzyme? Okay, any guesses?
Decarboxylase.
Perfect. The name will be decarboxylate, and the full name is pyruvate. There it was lactate dehydrogenase. Remember, here it's pyruvate decarboxylase. This is the enzyme. So through this enzyme, first we convert pyruvate into acetaldehyde, and this is the first step. Now, this acetaldehyde, which is now, again, it will be 2, right? So I'm writing 2 because there were 2 pyruvates. These 2 acetaldehydes will be converted into final product, which is ethanol. How many ethanol will be there? Ethanol. 2, correct? Ethanol, how many carbons are there in ethanol? Remember meth, eth, prop, bute, pent, hex. Methanol has 1 carbon.
Yes, sir.
It's also called formic acid. Then ethanol has 2 carbon. Propanol has 3. Butanol has 4. Write this. So ethanol has, it's again a 2 carbon molecule. I'm writing it 2C molecule. So which means we have not lost a carbon. So what we have done, we have converted an aldehyde to a ethanol, and in this process, remember this NADH which we got from glucose, since yeasts, so this is happening in yeasts, since yeasts cannot do at this moment, they cannot enter into Krebs cycle. So what instead they can do is this 2 NADH that they used, they will bring it here, and in the second step, these 2 NADH will enter and leave as 2 NAD plus, correct? So again, what's happening? Hydrogen is being removed. So can anyone guess the name of the enzyme? Yes, Mariam.
Dehydrogenase.
Yes. And what dehydrogenase?
Ethanol.
And what is ethanol? Ethanol is an alcohol, right? So it's called alcohol dehydrogenase. Specifically, it's ethanol dehydrogenase. You're correct, but we use the name alcohol dehydrogenase. Clear, people? And this here is your step 2. So it's a 2-step process from pyruvate to ethanol, and finally we make it. Okay.
Yes, sir.
Perfect. Anything confusing or anything you want to ask? All is clear. So write down few things about this process. One thing that they have asked is in the entrance was asked in AIMS was this enzyme here, pyruvate decarboxylase. Write down about this. The enzyme pyruvate decarboxylase. The enzyme pyruvate decarboxylase requires this one here. I'm writing about this. It requires thiamine pyrophosphate. Thiamine pyrophosphate as a coenzyme. So this enzyme cannot work alone. So it requires, this has been asked once, but I thought I should tell you. Pyruvate decarboxylase, which is used in alcohol fermentation, uses thiamine pyrophosphate as a coenzyme. And the good thing about this is, why this is asked is because this thiamine pyrophosphate is derived from vitamin B1. Derived from vitamin B1. Remember vitamin B? What is it required for? Vitamin B. Vitamin B complex. Vitamin B12, B1. People often take vitamin B complex after they are recovering from an illness, correct? Have you seen people taking vitamin supplements when they are recovering from illnesses?
Yes, sir.
Or also as a treatment of certain illnesses. Certain neurological illnesses also takes vitamin B, because vitamin B1 gives rise to this coenzyme, which is important in a lot of things. That's why. Okay. So everything is clear? Any doubts?
Yes, sir.
No doubts. Okay, so go through fermentation, both the pathways ones, and I'm giving you 3 minutes break. I'll get water for myself, and then I'll start the respiratory quotient. Okay. Ask any doubts if you have.
Yes,
See you
sir.
after a few minutes. Let's take a break. Okay. Hello. Am I audible? Mariam? Reba?
Yes, sir.
Yes. Welcome back. So for this part, everything is clear till now. Any doubts? No doubts?
No, sir.
Okay. So basically we have covered respiratory pathway and also we have covered fermentation. So if you see clearly and if you understand, fermentation lies between the aerobic and anaerobic, correct? So if you have to make a table or if you have to understand the difference between all the three, like compare between all the three, will you be able to do it? Comparison between aerobic respiration, fermentation, and anaerobic respiration. Okay, let's do one thing for you. Make a table, make 3 columns, write aerobic respiration, fermentation in the middle, and anaerobic respiration. Okay, before we move forward, let's just summarize it. Did you do it? I'll just do it verbally because I think you understand it. So did you make 3 columns?
Yes, sir.
First is aerobic, middle one is fermentation, and the right one is anaerobic, correct?
Yes, sir.
Yes. So aerobic, first point will be that it's oxygen dependent, right? Whereas for the other two, both for fermentation and anaerobic, it's kind of independent, right? Because fermentation also, whenever there is less oxygen, it decides to go into fermentation, correct? When it is less oxygen outside. So you can write in the other two, fermentation and anaerobic. In fermentation, you can write partially dependent on oxygen, correct? I think that's a better term.
Yes, sir.
Whereas anaerobic is oxygen independent. If this question comes in boards, then the second difference you can make on the basis of final electron acceptor. You know for all three. Tell me for aerobic, Mariam.
Oxygen.
Very good. For fermentation?
Acetyl.
Any endogenous organic compound.
Yes, sir.
Whatever it is, it's endogenous.
Electric.
If you just write endogenous organic compound, it's done. And for your anaerobic, nitrate, sulfate, fumarate, anything but not oxygen. Okay. Is it clear?
Yes, sir.
And then the third point, we can go to the type of phosphorylations that are happening. So in aerobic respiration, what happens? Both substrate level phosphorylation and oxidative phosphorylation happens, right? Both happens. Substrate level in glycolysis and oxidative in Krebs cycle and electron transport chain, correct? The third type of phosphorylation is photophosphorylation that only happens in photosynthesis. Is it clear?
Yes, sir.
Yes. So in aerobic respiration, both type of phosphorylation happens, both write down substrate level and oxidative. Tell me for fermentation. Only substrate level happens in fermentation. There is no oxidative phosphorylation happening in fermentation. Okay. And in anaerobic, this is the question. Which phosphorylation happens in anaerobic? Mariam.
Substrate.
Both happens in anaerobic. Now you will be little confused. Why both? Because there is no oxygen. So how is oxidative phosphorylation happening, correct? But oxidation and reduction can also, so remember in anaerobic also there is a inorganic molecule like sulfate or nitrate which is accepting electrons. So when it is accepting electrons, basically it's oxidizing something else, right? Not to the level as your oxygen can do, but when it is accepting electrons, it's oxidizing something because it is itself getting reduced. When oxygen was getting reduced, it was getting reduced into water, H2O, right?
Yes, sir.
So here also the nitrates and sulfates will get reduced into something, but the other thing will get oxidized, which is this NADH and FADH2, which is getting oxidative phosphorylized. So write down both substrate level and oxidative. But in anaerobic respiration, in front of that oxidative, just write without oxygen. So the name is quite confusing, but you understand the process, right? Is it clear now?
Yes, sir.
Okay. Now, canonically, on the basis of logics, we always think that we are breaking down things in respiration. So respiratory pathways or respiratory system is always called a catabolic system, correct? But actually it's not entirely catabolic. It's nowadays people are coming with, scientists are using better term for it, and they are saying that respiration is respiratory pathway is not entirely catabolic pathway, but it is a amphibolic pathway. Now the word amphi means
Both.
yes, amphibian, remember, which can live both on land and water. So amphibolic means, bolic means metabolic, so both catabolic and anabolic. So write down amphibolic pathway means which has both aspects, both catabolic and anabolic. And why do we do so? So this is one figure that I have taken from your NCERT. And this talks about everything that we have in our body, which we can break down or we can put into some pathway to produce energy. So the first thing we studied about majorly was carbohydrate, in which we studied about glucose. See here, right? Now glucose goes into this pathway, which is glycolysis. And finally, so this is, remember this step of glycolysis there, where one partner is acetone phosphate and another partner is aldehyde?
Yes, sir.
The fifth step of glycolysis. They have just shown this for a reason. And then finally it reaches to pyruvic acid. So this whole pathway is glycolysis pathway. And it ends here. And then it goes into mitochondria, gets converted into acetyl-CoA, and that enters into Krebs cycle and CO2 is released. So for carbohydrate, I think you this whole thing, which is going through this, it's clear to all of you, right? Everything is clear. Now, apart from carbohydrates in our body, we also have fats, right? The second thing is fats. Is it clear? Fats are made up of two things, fatty acids and glycerol. Now, this glycerol can easily go into dihydroxyacetone phosphate. It can become dihydroxyacetone phosphate through certain pathway, which they have not basically told you about, but it is possible. Glycerol can directly enter this pathway at this step, right? It can become dihydroxyacetone phosphate, which can get converted into glyceraldehyde 3-phosphate and then continue in the glycolysis pathway and then go into Krebs cycle. Clear?
Yes, sir.
The fatty acid component of fats, and why I'm telling you this, remember I told you, if a person is starved for a very long time, suppose a person is stuck on an island or something and there is no food availability and the person will die starving. So death through starvation goes in a phase. Remember, first we will deplete our carbohydrate reserves, right? Sugars, glucose. Then we will come to burning of fat, right? Whatever fat we have in our body, we'll start burning it, taking it out, correct?
Yes, sir.
And also the proteins. The third thing that, our own muscles start eating our own muscles, like our own body starts eating our muscles. So muscle starts degenerating because proteins are made up of amino acids. So these fatty acids and amino acids, both fatty acid can directly enter the pathway at the level of acetyl-CoA, the pathway that you just studied. And amino acid can enter the pathway by converting into pyruvic acid. Then pyruvic acid will convert into acetyl-CoA, and this thing is clear. This is in common, correct? So there are three major steps. One is this dehydroxyacetone phosphate, where glycerol can enter, through which and fats can get metabolized. The second is pyruvic acid, at which amino acids can enter at this level. Third is acetyl coenzyme A, at which fatty acids can enter. Is it clear?
Yes, sir.
Now, when it comes to entering, it can also be taken. So acetyl coenzyme A can be taken away from this pathway to form fatty acids when it is required in the body. Suppose someone's body requires fatty acids, a lot of fatty acids has to be made. So from where you can take the raw material? Right from your respiratory pathway. So you take extra acetyl coenzyme A from the respiratory pathway and put it in fatty acid synthesis. Is it clear?
Yes, sir.
Similarly, when you have to make proteins, you can take pyruvic acid which is extra pyruvic acid from your respiratory pathway and give it to the formation of amino acids. Is it clear?
Yes, sir.
So can you now appreciate that respiratory pathway is not only about catabolizing and breaking down things, it can also contribute to making up things, which is anabolic, which is called anabolism, anabolic in nature. So that's why nowadays we consider respiratory pathway as amphibolic pathway. Is it clear? So this question came in board, why justify a respiratory pathway as an amphibolic pathway. So will you be able to do it now? For
Yes,
that,
sir.
open your NCERT. I was just going through it, and I think what you have to open is, I think page number 235. There's one paragraph. Go to page 235. Read this paragraph, which talks about the amphibolic nature of this pathway. Okay. And you can make your answer from there. Is it clear? So whatever I explained you just here is written in that paragraph. Okay. Cool.
Yes, sir.
Okay. What was here? So if you have any doubt, ask me tomorrow. Read this paragraph, page 235 from NCERT, and I think everything else is clear. I have explained. So there's one thing that we have to finally, the final thing of today is your respiratory quotient. What do you understand by respiratory quotient? Remember, we studied something like this. I'll tell you where we studied it. Respiratory quotient. Let's first define it. Write down the definition. RQ or respiratory. Okay, it has one more name which I have seen in some books. It's sometimes also called respiratory coefficient. Both are same thing. Okay. So whether it's respiratory quotient or respiratory coefficient, it's the same thing. So write down. Respiratory coefficient is defined as the ratio of moles / volume. You can use both. The ratio of moles. You know moles by now. In chemistry, you must have read about moles, molality, normality, formality. Have you studied all this or not? Okay. So moles or volume of CO2 produced to the moles or volume of oxygen consumed. It's the ratio of moles or volume of CO2 produced to the moles or volume of oxygen consumed during complete oxidation. During complete oxidation of a metabolic fuel. Anything which we can metabolize of a metabolic fuel, we call it metabolic fuel. It can be glucose, malic acid, proteins, fats, everything. Sorry, what did I say, Mariam? Can you just repeat? It is the ratio of moles or volumes to CO2 produced to the moles or volumes of oxygen consumed during complete oxidation of a metabolic fuel into CO2 or water, into CO2 or H2O. Is it clear? So you can define it as, the formula is moles or volumes, whatever the question gives you, CO2 upon moles volume of oxygen, CO2 produced to oxygen consumed. Is it clear, everyone?
Yes, sir.
Now, let's just do this one simple thing. So if I give you so always these questions will be given as chemical reactions. So whenever we break down a glucose, what is glucose chemically? Can anyone tell me? Formula for glucose?
C6H12O6.
C6H12O6 plus we give oxygen, right? And we break down this into CO2 plus water. Can you balance this reaction, Mariam? And here the metabolic fuel is glucose, which is a sugar. Can you balance this reaction?
Yes,
Yes.
6 CO2 and 6 H2O.
6 CO2
and 6 O2.
and 6 O2, right? So now, according to this, if we want to calculate the RQ, then what is the total CO2 produced? Let's say 6.
6.
And total O2 consumed 6, correct? You can
Yes,
either
sir.
take O2 or water, but you have to take any one. Sorry, you have to take CO2 because water is not utilized, is not taken for this reaction, but water can be the end product. Here CO2 and water both are the end products, but when we will figure out the RQ, we will take CO2 and oxygen. So what is the RQ? 1. Do you understand? With the simplest equation. Is it clear? Is it easy enough to understand?
Yes, sir.
Yes. So now I'll just dictate you, write down certain RQs which you can remember, or also you can find out through this way. Should I give you one more question which you will solve? Let me give you for
Yes, sir.
malic acid. So write down malic acid and you tell me the RQ for malic acid. Okay. RQ of malic acid you have to find. Now I'll give you the formula. You have to balance it and find the RQ. Okay. Malic acid is C4H6O5. And we are in the presence of oxygen converting it into CO2 plus water. So quickly just balance this whole is O5. This is malic acid. Oh, sorry. Let me write it clearly. C4H6O5. Can you balance and find the RQ quickly for malic acid? Mariam, are you doing it?
Yes, sir.
Yes, good.
Sir, 4/3.
Yes. So what is the RQ?
1.3.
Perfect. Good. Should I give something slightly different? It's another question from another exam. You want to do more? Just one more?
Yes, sir.
So this time we are finding for there's something called palmitic acid. You know where is this present? Palmitate or palmitic acid? Body lotions, cold creams. How many of you apply body lotions or cold creams?
Yes, sir.
So for palmitic acid, you are going to find out. Okay. Now the formula. So I'm giving you this. C16H32O2 because it's an acid. So you remember if it's an acid, this 2O will be COOH. Remember?
Yes, sir.
So again, in the presence of oxygen, we are converting it into breaking down into CO2 and water. Now do this. It's not tough conceptually, but just numerically. So let's see if you can find the right answer. Tell me the RQ for palmitic acid. Difficult.
Sir, 0.6. 0.696.
0.7. Okay, just convert it. Perfect. So the RQ
Yes, sir,
for
0.7.
palmitic acid is 0.7. Now there is one rule that you should remember. You can write down after this that for most of the common fats, okay, which is like palmitic acid or palmitate is 1, the RQ is around 0.7 always. Write down RQ is 0.7, is around 0.7 for most of the common fats, and about 0.8 0.9 for proteins. Okay. And what it is for carbohydrate? For glucose it is 1. For glucose it is 1. So for carbohydrate it is 1. For proteins it is 0.8 to 0.9. And for most of the common fats or this palmitic acid, it's 0.7. Okay. What it was for malic acid? 1.3. Okay. And just remember for one important oxalic acid. For oxalic acid, it's very high, so you should just remember it. Sometimes they ask it in the entrance. It's 4.0. So it's highest for oxalic acid from the substances which are commonly metabolites. Okay. So carbohydrates 1, protein 0.8 to 0.9, fats around 0.7, malic acid 1.3, and your oxalic acid 4. It's clear, Mariam?
Yes, sir.
Clear? So respiratory quotient, if they ask you for any metabolite, they will just give you the formula. You will be able to find the RQ.
Yes, sir.
Very good. So this marks the end of respiratory system. So tomorrow's class will be the wrap up for plant physiology. There are certain important things that I have left in plant physiology to bind from the hormones chapter and also from some other chapter. So tomorrow class we will try to wind up with the syllabus, and if you want to ask something about your exams, you can do it. Okay. And tomorrow is also your viva. So first hour will be the viva and the next 2 hours will be the class. Is it clear? Please
Yes.
inform others. I think Aisha could not join back. So Mariam, will you please inform Aisha?
Yes, sir.
If possible, also share notes with her. Okay. Okay. So if you have any doubts, I'll take the doubts. Otherwise, I'll just declare the class. Any doubts?
No, sir.
Okay. Then see you tomorrow. Have a good night.